right, well, thanks for the invitation to present here today. I'm going to be talking about some work done by the Plant Reproduction Working Group on a paper that came out December 31st. So somehow right before New Year's or right in the middle of the holidays. So I'm happy to talk about it here. Community synchrony and seed production is associated with trait similarity and climate across North American LTER sites. And I'm Jalene LaMontagne. And I just moved actually to the University of Missouri St. Louis and the Missouri Botanical Garden. And so I wanted to start with just telling you a little bit about one day recently, I was walking through the Missouri Botanical Garden and the sign caught my eye as I was going to my office that said Victory of Science. And we sort of zoom in on that sign. It says the Victory is the Roman goddess of success in all things in victory and war. Note here her sword is set aside so that she is not depicted in war. The inscription reads, the victory of science over ignorance. And I just thought that was a lovely sentiment. And we all work to pursue knowledge and keep going. So the work I'm presenting today is this paper that came out in Ecology Letters, and everyone listed here is a member of our synthesis working group, and the co-PIs on it are Miranda Redmond and Elizabeth Crone, and all those happy folks during our first visit. So we were funded in early 2021 and didn't have our first meeting in person for quite a while. And with this work, we're looking at this scientific problem that seed production in perennial plants is highly variable over time, and I'll describe some of those patterns in a moment. But the drivers of these patterns are still poorly understood. But occasionally, we see these really large reproductive crops occurring on species. Here, these are all conifers, but it extends beyond that. And so mass seeding is defined as the synchronous production of highly variable seed crops by a population of perennial plants. And in this figure, you can see that on the x-axis, we have almost 30 years of data. And this is showing the mean number of cones produced per tree. This is for white spruce. And the pattern here to note is that many years are low reproduction. And then every once in a while, there's this really large resource pulse that happens. And we refer to that as mass seeding events. And for this species, what that looks like in the fall is that the trees will just be covered in these cones that have dried and then release the seed. And when we look at cones, they're comprised of all these scales, and there's room for 2 seeds at the base of each scale. So it's a massive amount of seed that's produced and also potential food. And in some regions, we can actually detect these events using satellite imagery. That's what this reference is here. And so this is a phenomenon that occurs in a wide variety of species of perennial plants. So acorn production in oaks, this occurs obviously in conifers, and then even some bunch grass studies that have been done in New Zealand. There are a number of hypotheses about why would species have this pattern of reproduction. And so a couple evolutionary hypotheses include the seed predator satiation hypothesis, that the idea is that if a population of plants just produce the same amount of seed every year, then those seed predator populations, either birds or small mammals or insects, would just increase their carrying capacity in order to consume all of that seed. So by having years where there's multiple years of low reproduction and then this pulse in reproduction, that could satiate those predators when there's that pulse that occurs. Another hypothesis is the pollination efficiency hypothesis. So this pattern of highly variable reproduction over time is common in species that are wind pollinated. And so if individuals were just randomly reproducing every year, then there wouldn't be very good pollination and very efficient seed set. So by individuals synchronously investing in high levels of reproduction, there'll be better quality seeds. And then proximately, how these individuals and populations are signaled to reproduce is thought to be weather cues. So hot and dry conditions during the time of bud differentiation is common in many temperate regions anyway. And so there's been a lot of work done on just one or a few species at a time, and at just one or a few sites historically. So we wanted to take a different approach because we were thinking about species don't exist in isolation, but they exist in communities. And that there could be advantages to this idea of multi-species synchrony. So if different species were completely out of sync, then that seed predator population would just increase. So if they were synchronous, then there would be more effective satiation of seed predators, so keeping them low and then satiating them. However, there could be disadvantages to that multi-species synchrony. So maybe there'd be competition for suitable sites for germination if there's high levels of reproduction occurring across species at the same time. And then we can also think about environmental drivers that influence woody plant reproduction. And many of them tend to be similar, these cues for synchrony and the role that they play. So the objectives of this paper were to quantify synchrony reproduction sort of at sites, but we looked at it sort of overall and then within each of multiple LTER sites. So we just want to know how synchronous are species that co-occur. Then we wanted to test for relationships between synchrony with trait and phylogenetic similarity, and then look at how climate conditions are related to levels of synchrony as well. So we did this using LTER data. We focused on woody plants. To be included in the study, there had to be a minimum of 10 years of data. The data on reproduction had to be identified to species because for this work, we also looked at species level traits. And so anything at the genus level or above would not be sufficient for that. And data could be either visual counts on individual plants, or they could be seed traps. And to be included here, there had to be data from at least 2 species because we're interested in this idea of cross-species synchrony. So synchrony and reproduction over time by co-occurring species. And you can't do that if you only have 1 species. So we ended up with data from 7 LTER sites across a large latitudinal range. So we have Loquillo, Coweta, Harbor Brook, Cedar Creek, Sevietta, Andrews Forest, and Bonanza Creek, and a large longitudinal range. And really different climate conditions here too. So mean temperature ranges, from almost -3 degrees Celsius to almost 22 degrees Celsius. And so there's some things that we found here that we couldn't have detected had we only looked at one individual site. So that's an advantage of the LTER network. And in total, we included data on 103 woody plant species. And these sites included a range of biomes from tropical rainforests in Puerto Rico to dryland areas in New Mexico, where there were only 3 species of woody plants, of course, in comparison to Puerto Rico, which has very high species diversity, and, up into the boreal as well. So with this, we also looked at the species traits. And so we had 11 different traits for all 103 species that we compiled using the TriPlant database, other literature, expert knowledge, and we also looked at phylogenetic relatedness as well. And then for part of our analysis, we either said, okay, do the species for a particular trait share the trait? If so, it's a 1, or is it different for that pair of species? And there's a list of all of the traits that we included. Most of them are categorical, and then we also included similarity and seed mass, and then phylogenetic similarity as well. This just summarizes the different LTER sites. So the 7 sites, the range of biomes that included, the range of species that occurred at these different sites. So again, ranging from 3 species to 62 species. And this is the number of species that we had data for that we could use. So at some sites, they might have had oaks, but they only had it at the genus level. So that's not included here. We also have the number of traits that we had some variability in them. And this is also taking into account phylogenetic similarity. So they all had that. Because at some sites, there wasn't much variation in the traits that the species had. And then at other sites, there's a lot of different variation that's there. You can see the number of years of data of overlap. So the way that we did this analysis for synchrony is you have the time series of the 2 co-occurring species, and then we said, okay, well, how much of their time series overlaps? And they had to overlap for a minimum of 10 years. And then we said, okay, well, how synchronous are those? Is that pair? And then we did that for all pairs of co-occurring species at sites. And at some sites, we had up to 58 years of data to look at synchrony. And then I'll also mention this number of plots here for the climate data. Those were done not just one for the LTER site, if there's multiple plots that existed. And so you'll see a bunch of data points in a figure a little bit later on that's representing those plots. So to look at objective number 1, we wanted to quantify synchrony in reproduction. And so again, I keep stressing this, that we're only interested in synchrony of co-occurring species at the same site, not comparing species in Laquillo to species in Bonanza Creek or anywhere else. We didn't care about that. So this is within site synchrony, again, minimum of 10 years of overlap, and the average number of years at the sites went from 17 to 41 years. And then in order to do this, we just compared the observed synchrony to taking each of the time series that actually exist, randomizing those, and then said, okay, well, how synchronous are they? And this is what we found. So this is on the x-axis is the cross-species synchrony, so the synchrony between pairs of species, and it's a density plot. And so the randomized synchrony is what we would expect. It's centered around 0 because it's just all random. And what we found that the observed synchrony between pairs of co-occurring species was significantly shifted to be synchronous, but also that there's a huge amount of spread. So we have some species, co-occurring species, that are very, very synchronous, right? Close to 1, and some species that are not synchronous really at all, or anti-synchronous. And then we looked at this within LTER sites as well. So here, the gray again is this our sort of null distribution from just randomization. And these are sorted from areas that are sort of the most arid are located at the top, and then sort of decreasing aridity in terms of climate water deficit. And we could see that some sites like Bonanza Creek, it's shifted to be positive. And this is a set of just 4 different species in their time series of reproduction. And we can contrast that to Andrews Forest, which is shifted to be very positive. All the species here are conifers. And when you look at the Andrews Forest data, just the raw data, it looks to be much more synchronous. You're getting, synchrony and when there are some of these pulses of reproduction compared to Bonanza Creek, that these lines are just sort of a little bit more all over the place. So that's shifted to be a little bit lower synchrony. So we showed that there was more synchrony than we'd expect just by random chance. And then for our second objective, we wanted to look at a test for relationships between synchrony with trait and phylogenetic similarity. And what this figure is showing is each symbol is representing an individual species and where it exists in sort of trait space. And it's color-coded by the LTER site. And this is where I sort of want to again, state this thing of, because we used data from the LTER network and across multiple LTER sites, we encompass more trait space than one could if you were only looking at one individual site and then going to draw conclusions. And that, I think, is a very important point to make and a strength of the LTER network and synthesis. And then for the analysis of, okay, well, are species that are more similar in their traits overall more likely to be synchronous? And the answer is yes. So the black line here is showing the overall patterns. Each of the symbols is an individual pair of species, what their trait similarity is, and their cross-species synchrony. I'm going to come back to that purple declining line in a little bit. And then what we wanted to do is look at individual traits and say, okay, well, how is this trait related to levels of synchrony for co-occurring species? And so this is an example, and my bar thing is in the way, so I totally can't see which one it is. But the trait status is a 0 if they're different from one another, and it's a 1 if they have the same value of that trait. The next slide, I have one that's zoomed in, and I'll go into more detail. But this is showing, and then the y-axis is the cross-species synchrony. So in this case, and because this is colored green, the species that have the same trait, regardless of what that trait is, but when they're the same, they're more synchronous than a pair of species where they have different traits. And we did this for a whole bunch of different species. If it's in gray, it wasn't statistically significant. If it's shown in green, then it is. And we did this for a whole bunch of species. And this is using multiple regression on distance matrices. And we did this both for seed mass similarity, which was not significant, and nor was phylogenetic similarity. So phylogenetic similarity did not play a role in the level of synchrony between a pair of species. So here's an example of pollinator code. So they could be pollinated by wind or by animals. And this is showing if they are both pollinated in the same way, they are more synchronous than if they have different pollinators. And then we can ask, okay, well, what are the pollinators and how does it matter? So if they're both animal pollinated or if they're both wind pollinated, they are more synchronous than a pair of species where one of them is animal pollinated and one of them is wind pollinated. And this may be due to, well, if you have animal pollinators, you don't want to have that erratic pattern of reproduction. Meanwhile, another co-occurring species would have an erratic pattern of reproduction, so their synchrony would be lower. We could also look at deciduous and evergreen. So again, if they're the same, if they have the same trait, they're more synchronous than if in this pair they're different. And in this case, what we find that if both species in the pair are evergreen, they are more significant than if any deciduous species is involved at all. So it's not always just, is the trait the same or different, but sometimes it's also a little bit more looking at those traits as well. And then our third objective was to look also at sort of the climate of the sites and relate that to synchrony. So this is the same slide that I showed you before with the LTER sites, organized by climate water deficit. So from the highest climate water deficit at Sevieta to the lowest with Laquillo and Coweta. And now we're going to look at the relationship between that climate water deficit and overall levels of synchrony between pairs of species. And in this case, this is where the 44 number comes in. So there's multiple, some of the sites are quite big. So there could be different values of climate water deficit that are used, whereas some other areas like at Cedar Creek, the sites used there were close enough together that they only had one data point for climate. But what we find is that more arid areas have greater synchrony in seed production. And then we also just wanted to look overall like, okay, well, we have data on trait similarity, climate water deficit, actual event for transpiration, and phylogenetic similarity, and overall, what are the important factors here? And then in driving the levels of synchrony, we find that trait similarity was significant, and also climate water deficit was. But phylogenetic similarity, this is quite an interesting result, was not a player in the level of synchrony between species. So to summarize, we found that cross-species synchrony is greater than expected by chance, that species pairs with greater trait similarity had higher cross-species synchrony. And I said typically, because if we look at this figure at the bottom in the middle, it's Sevieta that actually goes down. So this is an arid site. There's only 3 species of woody plants that exist. And for the species that had higher trait similarity, they were less synchronous than species that had lower synchrony. And maybe that's due to the stressful environment that these species are in. And again, an insight that we wouldn't get if we just looked at any particular site, or if we only looked at Sevieta, our conclusions would be different than using the network of sites. And again, driving home that the LTER synthesis approach provided this wide representation of species traits across a wide variety of biomes. So some of the implications of this work is giving new perspectives about mass seeding synchrony, looking between co-occurring species. And through this work, we suggest that these pressures exerted by seed predator populations, these seed predator populations, they're not necessarily interacting only with a single plant species. They're typically generalists, but they're interacting with whole plant communities. And that while mass seeding has this definition, about temporal variability and spatial synchrony, by populations of perennial plants, that these communities could also matter when we think about cascading effects of mass seeding in ecosystems as well. And along with this, we published an LTER data paper that came out in Ecology in January, co-mass, so community masting, and this is the harmonized seed production data. And so it includes everything that's in the paper that I talked about, and it includes, some other sites that didn't fit into the requirements, and some species that also didn't fit into the requirements. So I think there's about 140 species in this data set, and it's available for people to use. And with that, I'll acknowledge all the US LTER sites and everybody who collected and contributed data, because obviously none of this work is possible without them. The National Science Foundation. I mean, again, this is without funding for the LTER sites and the LTER network office and the synthesis working groups, none of this work would be able to be done. Also, a number of the LTER sites here supported by the USDA Forest Service as well. Of course, NC's LTER network office, you guys are awesome. And all the working group members, because they're not here, so I will acknowledge them as well. And thank you for listening. And there's my contact info at the bottom. Thanks, everybody.